Thank you very much for the invitation and the introduction. You know, I do have an MD also, and I'm sorry about that. Everything, no matter how many things people do in their lives, though, there's maybe one thing you really are. And I got my training as a physiologist. I think what I really am deep down as a physiologist. I know everybody here, though, shares, regardless of the system that you work on, whether there's a clinical or preclinical or fundamental basic science challenge, there are some common themes that we all face, and one of those is to try to keep the system as intact as possible while still achieving high precision control and exchange of information with elements within the system. And so if there is a unifying theme, that's it. The problem, I think, is particularly acute in psychiatry. This is the kind of patient I see in the clinic, just a half day a week, but this is epidemiology, which I think is not widely appreciated. Surprising to me, first time I saw it. The red pie slice is the fraction of worldwide disability that's attributable to neuropsychiatric disease in terms of years of life lost to disability. These diseases are chronic, often fatal, poorly understood, hard to treat, and of course this is an incredibly challenging system. The approach that we've taken is to use light to try to study individual cell types within the brain, to seek the activity patterns that control behavior, and to try to apply these approaches to biology, both in health and disease. And what I'll share are two technologies. You've heard a brief introduction, but they share this theme of trying to keep the system maximally intact while achieving high resolution interaction with the system. Now, this may turn out to be one of the strangest introductions to a physiology talk that you've seen, but I'm going to start actually by talking about chitin. This is one of the more common biopolymers. It's a major constituent of, for example, the wing of this moth that I caught in my office when I had a brief moth infestation. It's an incredibly durable, robust polymer, and it's made from very simple sugar-based monomers, which, as I was thinking about the problem of keeping the brain intact while being able to observe structure, I thought, what if we could make a durable endoskeleton within the brain? What if we could trigger polymerization of something rigid and durable, perhaps in a targetable fashion to a particular subset of elements within the system? And I was reading and looking into this. This was a number of years back, 5 or 6 years back, and one thing I noticed was that the monomer of chitin actually is present in neurons. It's used for other reasons, of course, but the thought I had was, well, what if we introduce a chitinase that would polymerize it? We could create, perhaps within a targeted subset of elements within the brain, a endoskeleton that would allow the structure to be studied, perhaps even filling the axons, fine branches of the dendrites. And of course, there would be other versions of this. And I had this is one of our laboratories. This is a picture taken by one of my postdoctoral fellows, Aaron Andelman, when there was a rainbow over the building. It's usually not this dramatic, but this is where the early work was done in what later ultimately became Clarity. We looked at chitin. We looked at cellulose production. Cellulose also is made from sugar monomers, which are present, although its biosynthesis is a bit more complicated. Keratins also were a possibility. This is a slightly different strategy, but you could build, in theory, durable endoskeletons by creating keratin polymers within cells. So this was the sort of thing we began to tackle, and some things sort of worked early on. And so one can, for example, express keratin 8 and keratin 18 side by side in the same cell. Here coupled to a yellow fluorescent protein, it does indeed, the polymerized keratin can indeed fill the cell. And if you express either the keratin 18 or the keratin 8 alone, here fused to two separate fluorescent proteins, you do not get polymerization. This is known from earlier biochemical work studying the process of keratin filament formation. Now, not only did it fill the processes, but the cells were durable. Normally, if you have just a cell and you do a very simple form of lysis, hypotonic lysis, the cell will fragment and you can see this sort of process happening. But the cells that have produced this endoskeleton within themselves, although they are, of course, dead and lysed, they leave behind this endoskeletal structure. So the idea was maybe we could do anatomy in a way that would allow us to visualize in the intact brain the structure of the targeted circuit. Now we had a small group working on this. Started with Viviana Gradinaru and Sharda Ramakrishnan in my laboratory, and then we started to try to move into three-dimensional systems, and I hired Kwang-Hoon Chung, an extremely talented chemical engineer from Georgia Tech who joined the group, and he started building these three-dimensional collagen networks that we basically cubes that we would seed neurons and glia into that were expressing these keratin filaments. And then we even actually got to an intact brain. We were introducing an adeno-associated viral vector expressing these keratin proteins, and it actually worked. We could create a cell-filling durable endoskeleton within neurons, even in vivo. Now, one thing we noticed as we started to want to try to then take a better look at these endoskeletons, and we wanted to try to clear away everything that was around them so we could visualize the endoskeleton better. And we wanted to provide support while we were clearing things away. And in the process of doing that, as we were adding in various kinds of support structure and adding in hydrogels to support the structure, much as we had been doing in vitro, it gradually became apparent that we didn't need the endoskeleton anymore, that the exoskeleton effectively had become so good. And this ended up becoming what we now call clarity, where one doesn't need to actually create the endoskeleton at all. So it's an interesting story. Sometimes science takes you in funny directions. And the end result, this is a mouse brain where YFP is expressed in Thigh-1 projection neurons. This is a never-sectioned mouse brain. We have thalamus down here, hippocampus here, and above that we have layer 5 of cortex. Here we're flying over the CA1 layer of the hippocampus, important in aspects of memory and affective regulation. Now looking up toward the pial surface through the layer 5 neurons. These are the apical dendrites rising up to the pial surface. So this clearing effect and support effect was achieved with 2 separate steps of the clarity process, and I'll tell you how that works now. Now there are, it's been a long-held goal to try to make tissue transparent, and many pioneering methods have been targeted toward this. One thing that sets clarity apart is perhaps the speed and completeness of the clearing, but more importantly the fact that it, by removing the lipids, actually leaves the structure susceptible to interrogation with macromolecules for labeling, including antibodies and in situ hybridization probes. The way it works is first, we can take actually even previously fixed tissue, including from brain banks, but one can also perfuse the brain, perfuse an entire animal transcardially with the monomer solution. There are different ways you can do this, but we use most commonly acrylamide-based monomers along with cross-linkers, bisacrylamide, and thermally triggered initiators of polymerization along with formaldehyde. So it's a mix, a cocktail, but in the cold, no polymerization happens until the monomers have had a chance to penetrate fully through the target tissue. Then, when we elevate the temperature, the process of embedding really starts, the thermally triggered initiation begins, and the hydrogel meshwork forms from within the tissue. It's built from within the tissue. This couples to proteins and nucleic acids by virtue of the amide linkages, which they have with formaldehyde, but lipids typically are spared from this cross-linking process and become susceptible to removal. The structure is quite durable and highly cross-linked, and as a result, one can be fairly vigorous with how the removal of lipids happens. We use strong ionic detergents for a couple reasons. Strong ionic detergents allow us to achieve a active removal of lipids, where we can effectively electrophorese the micelles out of the tissue. This speeds up the lipid clearing process by orders of magnitude. Also, though, these ionic detergents do not create as severe a quenching problem of fluorescence as hydrophobic detergents will do. The brain or tissue is put into a very simple device, a electrophoretic clearing chamber, and this process takes a few days, with the end result being like this. And fine structure can still be seen. These are spines on a dendrite in one of the Thigh1 YFP mouse brains. This is from a different transgenic mouse. It's a Thigh1 Channelrhodopsin YFP mouse line to show a membrane protein, in this case a Channelrhodopsin. But these are axons in cross-section, illustrating that although the lipids are gone, the physical form of what is left behind can be visualized and the structure interpreted. And then, because the lipids are effectively removed, this is the first step in what allows in situ hybridization or immunostaining to be carried out, even through multiple cycles, because of the robustness of the cross-linked tissue hydrogel hybrid. Small epitopes, small things like single amino acid neurotransmitters, GABA, can be visualized, co-stained for parvalbumin here. This is a D2 dopamine receptor in situ hybridization in striatum, picking up about half of the cells as expected. The blue is a DAPI stain. We looked at the protein loss, quantifying the protein loss into the solution in the course of this process and comparing it to conventional histology methods. There is, even with conventional detergents like Triton X100, 0.1%, you have a significant amount of protein loss, probably because you have just fixation here, but fixation plus hydrogel embedding here. There's even less protein loss in the clarity case, despite the use of a more stringent detergent, in this case SDS. If you do SDS alone without the hydrogel embedding, this is why you don't use SDS for conventional immunohistochemistry. So promising early results, and the question then became, could we apply this even to clinical samples from brain banks, capitalizing on the fact that you're not limited to looking at transgenically introduced fluorophores, but could stain for native antigens. And so here's a brain area 10 from a 7-year-old child who happened to have autism. This was a sample where we were able to stain for myelin basic protein to identify projection axons, co-staining for tyrosine hydroxylase and parvalbumin, a neurofilament and GFAP, somatostatin and parvalbumin. And we could actually trace and follow neurofilament labeled axons over substantial distances. This again is from the brain area 10 of the human patient, where the stain is for neurofilament. And because of the volumetric continuity, one does not have to carry out serial section reconstruction and alignment, and it's possible to be fairly confident of the three-dimensional structures that might be present in the intact tissue. Now, the multi-round immunophenotyping is interesting. This is a labeling for tyrosine hydroxylase in red superimposed on the EYFP transgenic label in this mouse. The construct is robust enough that a stringent elution can be carried out, removing the label, and then one can come back with a different series of labels and restain the same tissue. Not only does the structure appear relatively conserved through this stringent elution process, as you can see in this, for example, in this upper left corner, the individual cells with their YFP label retaining structure, but one can come back for a third round of staining after 2 rounds of elution and restain for the first stained epitope. And anagenicity appears to be preserved, as you can see here. This is the same field of tyrosine hydroxylase labeled cells. So carrying out all of these, this is now back to mouse. This is a mouse hippocampus, multiple labels in a rendered volume. This is a never sectioned mouse hippocampus. And you can perhaps start to appreciate one value of this is not only the multiple labeling, but having everything intact within a single data set. One can begin to appreciate joint statistics, relationships among different elements within the same preparation. Labels that are consistent with understanding the genetic identity are important, but also activity history are possible. Staining for CFOS, for example, is possible. And that can be integrated with wiring information. That capability to image and the capability to stain with antibodies can be carried out in a fully intact mouse brain, which is illustrated here. This is staining for tyrosine hydroxylase in a completely intact mouse brain. Even a half millimeter or a full millimeter or close to the middle of the brain, 2.5 millimeters deep. One can pick up here, for example, the known but sparse tyrosine hydroxylase expressing dopamine neurons within the dorsal raphe nucleus, fairly deep in the brain. And the specificity looks about as expected. Here, tyrosine hydroxylase labeling is in red, and DAPI is in green to identify nuclei. And you can see the tyrosine hydroxylase expressing presumptive dopamine fibers investing the nucleus accumbens neurons, but sparing the anterior commissure, as would be expected. In the prefrontal cortex, you can see fine Th-positive fibers investing some cells, but not others. So there appears to be specificity and the ability to visualize fine processes, even in the intact brain. So then the question becomes, what sorts of things could you do with this? One problem off the bat is the very large data sets. How do you handle them? How do you quantify things? How do you compare them? I'll get to that in a moment. Of course, one value of the large data set really is that it's a global brain-wide data set. The animals can be put through, for example, behavioral tasks, and in a relatively unbiased fashion, one can look for patterns of activity that might be brain-wide or correlations in activity across the brain that might have been difficult to appreciate otherwise. And those might be linked with the right sort of label to wiring or connectivity information. But as you can also appreciate, this is just a movie. This may be interesting, but it's not really data. This is the big challenge we're facing. It's actually a common problem across, I would say, many laboratories, many investigators, many fields struggling to deal with data sets that are describing large intact systems. And of course, the problem becomes worse the more resolution you seek to achieve. Clarity is compatible with everything that light microscopy can do, for certain. In fact, it even is straightforward to visualize synaptic connections within the limits of light microscopy. One can see opposed pre- and post-synaptic markers like this synapsin and PSD95 juxtaposition. And because of the reduced scattering, resolution is maintained even quite deep within the tissue. And actually, you can do electron microscopy. This was surprising, but the fact that the proteins are left behind but retained in place actually preserves nanoscale structure. And here in a clarified mouse brain, where we've done a conventional EM preparation without a lot of optimization, it's still quite straightforward to visualize synapses with postsynaptic densities, presynaptic active zones, mitochondria, and myelin sheaths as well. Again, likely because the proteins are left behind. Still not beautiful EM images as expected, I think, with all the lipids gone, which are a major source of contrast. You wouldn't expect beautiful images, but you can see synapses, and so at the very least one could, for example, take a volume which had been studied with light microscopy and identify is there some ultrastructural logic that we need to understand about this volume. Sections can be taken, EM can be carried out, and synapses visualized. Another question might be, well, what is the ability to look at fine processes? A major challenge with many kinds of intact imaging is the bleaching that happens when we seek to do high-resolution imaging of large volumes. This is a standard confocal imaging of Thy1 YFP expression in hippocampus, and it's possible to track axons and dendrites, but an objection might be raised. This is a sparse, Golgi-like stain, and so you could say, well, this is a pretty easy preparation in which to carry out such fine process tracking. What if you had a more dense label? And essentially what we found is that within the limits of how high your resolution can be with light microscopy, you can do a pretty good job of tracking even dense labels. MAP2 is expressed heavily in almost all dendrites, and here in this MAP2 immunostaining, imaged at high resolution with a high NA objective, we can actually track quite well, even within a dense neuropil, individual MAP2 positive fibers. We'll fly through this volume in a minute. This doesn't mean that, as with any method, this is certainly not a guarantee that one would not lose track of or have a problem with resolution of adjacent processes. That always could happen. Okay. Big data problem, I think. Do I have the ability to restart it, or can you guys do that? I don't seem to have capability here. I could take questions while it's being... Okay. Let's see if we can advance that. Okay. Yeah, so I'll go past that. In fact, I'll skip past this movie just to be safe. Okay. Now, so there are a lot of interesting issues going on, how to optimize the process, and we started a online forum, which this was a day after we started. It actually triggered a lot of interest. This is the URL, if you're interested. This is actually turned out to be a really interesting community experience, and people create their own topics and post and discuss on them. And in addition to this, we actually have also a more dedicated resource site where we have very detailed protocols, inventories, parts lists, sort of analogous to what we did with optogenetics at our optogenetics.org site. The more detailed source of protocols information is at clarityresourcecenter.org. And there on that site we have things like how to build your chamber, detailed images of step-by-step assembly. And this is, people are picking up, this is actually a shot from Sam Wong's Facebook page where he has a little comment on something becoming clear. This is over a barcode. Here, Sheena Jocelyn's group, she picked a quote from H.G. Wells' The Invisible Man. This is a mouse brain. And then they started, this is Ule Keen's lab. There's a spinal cord over that original quote from Cahal. And then Paul Franklin and Sheena Jocelyn got really funny. This is a mouse brain pre and post clearing. So people have it working, but then the question is, what is, now that you can do this, how could you make it faster, better, and what else could you do? So the problems, I've alluded to the big data problem. That's a fundamental issue common to what many people are facing in different fields. Another fundamental limitation is the working distance of the objective, of course. We've used 3.4, 3.6 millimeter working distance objectives. If you flip the mouse brain around, you can image it completely intact, just stitching together 2 imaging sessions. There are some companies, Olympus makes an 8 millimeter working distance objective that would allow you to visualize through an entire mouse brain. I believe that's a 0.9 NA objective, and Leica, I think, is making an even higher NA objective that will allow about 6 millimeters working distance. So that's a big step, and that's, of course, important. If you're willing to give up on high resolution, you can have even longer working distance, of course. So it depends on the resolution that you need. If you just need cell bodies, even more rapid things become possible. We're working on speeding up the process. The clarifying process can be accelerated in various ways. We've increased throughput even since the publications. We have chambers in parallel. You can do away with sinks, just have reservoirs of buffers, and so you can actually end up having banks of these clearing setups running at the same time. Staining, we're working on accelerating. The speed of imaging is a big deal. Now that you have an intact volume, how can you speed up the imaging? Confocal works well, but it has a problem that even though you're collecting from a point, you're illuminating your sample, and that can create a bleaching problem over time. This might be partially alleviated by light sheet microscopy, where there's a selective planar illumination, potentially reducing bleaching time. Of course, in many versions of this, you have a problem with z-axis resolution. Another possibility might then be two-photon line scanning with temporal defocusing. There are other possibilities for speeding up imaging. Another class of questions is, it's actually kind of interesting that you can infuse in to an intact tissue this monomer and build something from within it. It's a strange concept, but if you can do that, what else could you do? Could you, thinking really far out, could you introduce conducting polymers to create interfaces with the tissue that might be electrically active or that might be actuated in particular ways? Could you introduce enzymes? Since antibodies can get in, enzymes could get in. Could you do reverse transcription? Could you do RNA-seq within an intact volume? These are some interesting possibilities. If you set the porosity of the hydrogel, you can do this by changing your cross-linker concentration and the duration over which that process is allowed to proceed. You potentially could even create a setting where cells could be allowed to penetrate through the tissue, which would be interesting because proteins are present, of course, there and might be able to exert effects on proliferation and differentiation. So that's a brief overview of the clarity work. Now I'll shift gears and talk about some recent optogenetic work, if that's okay. And that, of course, is a different class of thing, but hopefully still interesting. This is the crystal structure of Channelrhodopsin. This took a long time to get to. It's hard to express these microbial proteins at very high levels, and we tried a wide range of different chimeras of Channelrhodopsins from different organisms to try to achieve higher expression levels, and one finally worked. It's a chimera between Channelrhodopsin 1 and Channelrhodopsin 2 from Chlamydomonas reinhardtii. And together with Osamu Nareki, we were able to come to this crystal structure, and it resolved a lot of interesting questions. A lot of people thought, how could a 7-transmembrane protein be a channel? It turns out it's got these two helices, transmembrane helix 1 and 2, that are tilted a little bit out from the rest of the structure, and a pore is created here. It is a dimer, two 7-transmembrane proteins attached to each other in the membrane. With a pore, and it's light actuated because of this all-trans retinal in magenta here, which is embedded within the protein, receives a photon and carries out a conformational change. And now that we have high-resolution information around the retinal, we can start to do intelligent mutagenesis to create altered spectral and kinetic and even selectivity properties given the proximity to the pore. And this is possible. We were able to take a Channelrhodopsin from Volvox carteri, a multicellular green algae. This one had an interestingly red-shifted action spectrum compared to the blue light-activated spectrum of Channelrhodopsin 2. And it can be driven out in the amber, 580 nanometers or so. But the currents were small, 100 picoamps or so, with whole cell patch clamp and culture. And we, over the subsequent years, since 2008, we've been working on improving the expression levels, and we've gotten about a 10-fold improvement now, culminating in something that we call C1V1. This is a chimeric Channelrhodopsin 1, Volvox Channelrhodopsin 1 protein with some additional mutations and trafficking modifications that generates really enormous currents, up to 2 nanoamps or so. in single cells. And how did we do that? Well, trafficking motifs to improve membrane trafficking is indicated here with this TS, the chimera, some point mutations. I won't dwell on the details. The end result, very high expressing opsin, still redshifted. In fact, we even reduced some of this overlap here, and can be driven with red light. Now, this one turned out to be useful for two-photon microscopy. This is a issue that a lot of people are having difficulty in achieving effective two-photon excitation of opsins, which of course is important if one wants to achieve single-cell control within intact tissue. And a number of people have made quite useful advances on this front, but one challenge had always been that the advances that were made were not really compatible with most people's two-photon microscopes, which carry out raster scanning. This C1V1 and a couple of mutations actually allow traditional raster scanning two-photon microscopy to achieve two-photon triggered spike patterns within neurons in intact tissue, in slices and even in vivo, as I'll show you in a moment. And we think we don't fully understand this. It's partly because of the redshift, partly because of high expression levels, and partly because of its off kinetics. We think that allows, its slightly slowed off kinetics allows the cell to integrate current over a slightly longer time and achieve action potential threshold. But we end up getting pretty good resolution. If we shift off the z-axis by more than 5 microns, we lose the spike in either direction. So you get about single cell resolution. We end up doing something a little different with our fluorophore. We've traditionally fused opsins to the fluorophore. That becomes hard when you're picking individual cells to excite because your dense tissue ends up looking like this. When we separate the opsin from the fluorophore, so the fluorophore becomes cell-filling instead of membrane-bound, it becomes much easier. That's a small technical aspect, but a very useful one. End result is we can now drive one cell but not another, and the other cell but not the one cell, as we wish, even in intact tissue. This is a very important panel. It's kind of buried in the paper, but I just want to draw your attention to it. What's plotted here is the jitter, the standard deviation of spike timing, as a function of the whole cell photocurrent elicited by the two-photon raster scan. And what you can see is that if you're above about 300 picoamps, your spike jitter is very low, on the order of 5 milliseconds or so, or a little more. And that is an important number to be aware of. This is good. This gets us in the range where we are, for example, faster than many spike timing dependent plasticity rules that are operative in intact tissue. But as you get to lower photocurrent levels, the variance in spike timing becomes greater. This highlights the importance of achieving high expression levels of opsins, but it also raises a caveat. You need to be aware that in the regime of the tissue where not only might expression level be low, but where light penetration might be low, and therefore photocurrents low, one could get a greater jitter of spike timing. And so that's an important thing to keep in mind, but you can certainly track fluorescence, and that predicts two-photon photocurrent quite well, so you can at least be sure or fairly sure of what your expression situation is and how that informs what your photocurrents will be. We've also, for the sake of time, I won't dwell on it, but we've been able to make bistable two-photon tools, also published in the same paper, and two-photon inhibitors, published in the same paper. And this is some work done in collaboration both with Rafa Justi and Mark Schnitzer, where we expressed one of these two-photon excitable opsins in vivo and did a whole cell patch clamp in neocortex of a living mouse. And here you can see moving off the z-axis and preserving single cell resolution even in vivo. So we think this will be a useful tool for people who wish to do single cell resolution work. I want to close with a couple recent vignettes on using optogenetics to control behavior in freely moving mammals. And we use for these efforts the fiber optic-based neural interface developed by Alex Aravanese and Feng Zhang in the laboratory back in 2007. We've used that for various kinds of behavioral conditioning over the years. I want to highlight one recent study which focuses on depression-related behavior, and another study that came out about the same time focusing on motivated behavior in the setting of behavioral challenge. So I see, the patients I see in clinic, I focus on two classes of patient. One is autism spectrum disease, and the other is treatment-resistant depression, which is a very interesting disorder, quite common and very serious, hard to treat. We have in animal models some things we can do. Of course, nothing quite fully matches the human condition, but individual symptoms can be modeled. This is an interesting one. This is the forced swim test. The animal's put into a cylinder that's about 2/3 filled with water, and it swims and tries to escape, and after a while it stops trying so hard. And this transition from an active coping state to a more passive coping state happens over time with some predictable kinetics. And that, forget about depression, forget about psychiatry, that in itself I think is interesting. That's an interesting behavioral state transition where an animal moves from an active coping strategy in a difficult situation to a state where action is not taken, where some assessment's been made at some level that action is not worth expenditure of the energy. And so what's going on in the brain while that's happening? We do know, of course, coming back to depression, we know that depressed patients tend to emphasize that transition. They often see in their hopeless-like state intense futility, pointlessness of action. This contributes to dysfunction and ultimately, in many cases, unfortunately, to suicide. But in the non-pathological case, it's interesting as well what's going on in the brain while this transition is happening. Now, the forced swim test traditionally is not a very high resolution, temporal resolution test, but Melissa Warden in the laboratory made it more rapid in its readout. She attached a magnet to the hind paw of the animal and an induction coil around the cylinder so she could pick up every kick the animal made with millisecond resolution, all the while having multiple single units recorded in the brain with tetrodes, and she was able to get that waterproofed in a low noise situation so she could pick up and resolve units, all the while also having optogenetic control, again with millisecond precision, over individual classes of circuit element within the tissue. So interesting preparation. Took her a while to get that together, as you might imagine, but it worked out in the end. She first validated it, found that the automated scoring tracked quite well with traditional manual scoring as expected. And then the first question is, well, what are the neurons doing? Particularly, she focused on, initially, although I'll show you some other regions, she focused on medial prefrontal cortex, where she hypothesized some of these decisions governing this behavioral state transition might be going on. If there is a region that's homologous to the interesting deep brain stimulation target of Helen Mayberg and colleagues, the subcallosal cingulate gyrus, where deep brain stimulation, it looks like it's therapeutic for depression, it would also be this region. So what are the neurons doing? Well, here are 3 epochs. This is the baseline epoch before the animal is put into the forced swim test. This is in the swim test, and then the animal is returned back to its home cage after the swim test. In red here is just one unit, one single neuron. And in black is the movement of the animal. And a coil was also put around the home cage so she could track movement before, during, and after the swim test. And this particular neuron in this rat was representative of what many neurons did, which was interesting. It was quite active even when the animal was not moving much in the familiar, safe home cage. So this is not, she's not in motor cortex. This is not a neuron that's reporting on movement. But in the swim test, that same neuron, and here, by the way, you can see over minutes, note the time scale here, 60 seconds, you can see the animal transitioning to the passive coping state or hopeless-like state if you're thinking in the psychiatry model. And this neuron seems to encode or represent those active moments all the way through when the animal is trying to escape, we infer. But this is not an evolution in the cell. As soon as you put the cell back into the animal back into the home cage, that reverts back to that initial pattern. So these are interesting cells. They seem to, their activity, they're not motor related, but they seem to represent a positive or active effort to escape a challenging situation. Many neurons are reflective of this pattern. In fact, there was asymmetry. This is a pattern of cells that were more active during the baseline than during the swim test, but during the swim test more active during the mobile than during the immobile epochs. The converse was very rarely seen, and the cells that seemed to encode this seemed to be relatively wide spike principal neurons. The presumptive inner neurons, narrow spike width, seem to represent less of that class of information. So these correlate with an interesting behavioral transition. Are they causal? Well, one thing you could ask is, does their activity change right before the behavioral state transition? And so this, of course, points to the value of doing many of these things together in the same animal at the same time. And indeed, for the single cell and then for the population, you can see a ramp up of activity just before the onset of mobility, as shown in the dotted line at time 0. That was seen more often than not, although not always. Still correlative, not causal, but then she could come in with an optogenetic intervention and test causality. So she introduced a channelrhodopsin under control of a chem kinase 2 alpha promoter to favor expression in excitatory neurons guided by the physiology result, and drove those cells with blue light, and here comparing a channelrhodopsin with a YFP control cohort. And the blue light indicates when the blue light stimulation was delivered, and unfortunately and disappointingly, very little effect was seen on the kick frequency, and this was showed up in multiple cohorts. So she then asked, did she have to resolve the population more? Of course, prefrontal cortex does many things, and so what she took advantage of then was what we call projection targeting. Microbial opsins can diffuse down axons and make them light sensitive, and one can transduce medial prefrontal cortex but then illuminate with a fiber optic the dorsal raphe nucleus, for example, is shown here. She looked at a number of different downstream targets, but here one can recruit these cells based on their connectivity, therefore being more refined in the population selected. And there she saw a very different pattern. Every time cells bearing that projection were driven, she saw increased mobility that was not seen in the controls. As expected from the recordings, but just to show you again here, this is not a motor cortex effect or a gross locomotor effect. In the open field test, driving those cells certainly did not increase gross locomotor speed. Other kinds of specificity were seen, for example, driving the cells that have a basolateral amygdala projection did not show this effect. And interestingly, cells bearing a lateral habenula projection showed the opposite effect. When those were illuminated, there was a small but significant reduction in mobility. And this lateral habenula is associated with other kinds of aversive or disappointment-related signals, and so it was not inconsistent with an emerging understanding of what this structure might be doing. So that's a brief overview of that effort, but you can start to see the possibilities now having freely moving animals where you can record, track behavior, and control all with millisecond precision at the same time in the same animal in relatively complex tasks is an opportunity both for basic science and for understanding the mechanisms of disease. I'll close with just a couple slides on a related project. We took those same kinds of tests, that animal in the forced swim test with high precision, and nothing that I mentioned was really a depression model, in the sense that the animals were not stressed, were not put into a depressed-like state. Well, it turns out you can relatively easily put rodents into a depressed-like state using chronic mild stress, or CMS. This is something we and many people do. It is a few weeks of non-painful but recurrent and continuous stressors that are unpredictable to the animal. And one thing we noticed, we did some recordings in the ventral tegmental area, source of dopamine neurons, for a number of different reasons. We were interested in hypothesizing that those might be altered. We did not see an overall firing rate change in the chronic mild stress or the non-chronic mild stress animals, but we did see different efficacy in spike bursting. So a lower percent of the total spikes were in bursts. The bursts were shorter in chronic mild stress with fewer spikes per burst. Now, to make the numbers show this, what actually happens is there are more bursts in the chronic mild stress, but they seem weaker, less organized, fewer spikes, shorter duration. So the VTA in this case seems to generate less robust spike bursts. So we then took the rats that we had made in the laboratory. Alana Witten and colleagues in the laboratory had made tyrosine hydroxylase Cre recombinase driver rats into which we could inject a Cre-dependent adeno-associated viral vector carrying an opsin and do this same automated forced swim test, still in rats, but in Th Cre driver rats, and deliver bursts of spikes to the dopamine neurons in the VTA and ask what happens. And what was interesting is in the forced swim test, every time those cells were driven, a consistent increase in escape-related behavior was seen, suggesting we could restore back this pattern of activity that had been missing and get an enhancement in escape-related behavior. And that was an effect seen in the forced swim test, but not in the open field. And I'll close with one slide showing really just the complexity. I don't have the answers as to what all this means, but just to show the opportunity that we have here. The high resolution means we can see what's the neural activity leading up to a kick, what's happening just before, during, and after a kick. We can look in different downstream structures, and it gets complicated. I'm just going to give you an overview of this. I don't know what it means, but I'll just show you the kinds of data we can now get. So for example, now this is recording in nucleus accumbens, which is a major downstream target of dopamine neurons in the ventral tegmental area. But the light fiber is in the ventral tegmental area. So we're driving dopamine neurons in the VTA, but recording units in the nucleus accumbens, a downstream structure, all the while the animal's in the forced swim test. Okay, so the first thing is you give a pulse of light, and what happens? Well, many neurons in the nucleus accumbens increase their spiking in response to light. That was a fairly common pattern. Another question is, what happens around a kick? If you synchronize, if you look at the spikes, and these are rasters for individual presentations, what if you look around each kick? Many neurons in accumbens do ramp up their activity just before a kick and peak right around the time of a kick. So in accumbens, there are neurons in this depression-related task that are representing the escape-related behavior. Other neurons seem to show the opposite pattern. No surprise to the systems physiologists in the room. Okay, now final point is what happens then if you compare what's going on in the dopamine modulated state where you're driving bursts of spikes in dopamine neurons, and you compare that to the light off case, the low dopamine case. And you look at what the neurons in accumbens seem to be representing about kick, about escape-related action. And we saw many different kinds of cells that were interesting. But what's interesting is some cells clearly changed what they were saying, what they were representing about the behavior, depending on whether or not you were in a high or low dopamine state, which was interesting. Some cells went from not really representing kick at all in the low dopamine case, where we're not driving bursts of spikes in the dopamine neurons, to very clearly representing kick. And these were interleaved trials. The cell would go back and forth to representing or not representing the kick. Other cells started off representing kick, and in the presence of the bursts of activity to dopamine went to not encoding it at all. Some cells switched. Actually, this was a rare phenotype, just one. Some switched their behavior and some stayed constant. The overall picture, though complex, though, is that the bursts of spikes to dopamine neurons can change the representation of action and accumbens. And we're still at the very early stages of trying to sort out what this means, just highlighting the opportunity now of having all these data streams present at the same animal at the same time. So that essentially is what I'd like to share with you. We have many other things, things relating to anxiety, substance abuse, social behavior. There are a number of opportunities, things we don't have time to dwell on, but I do think it's remarkable that, you know, we've been able to make some headway with these tools that come from microbial opsins and understand some of the most, you know, complex behaviors that mammals can carry out. So I want to pay particularly close attention to our colleagues who've been so crucial over the years. I mentioned Osama Nurecki, our collaborator on the crystal structure of Channelrhodopsin, which was took us a long time, but very much worth it in the end. Peter Hagemann, a collaborator over many years, working on aspects of structure function. Other collaborators along the way, I mentioned our team working on the Twophoton methods. We have a number of people who've worked together with us on appetitive behaviors, including Anto Banchi, Patricia Janik, Luis de la Sea, and others. And then many very talented students and postdocs. I just want to point to a couple people whose work I particularly spent time on. Melissa Warden, who developed the automated forced swim test, now starting her assistant professorship at Cornell. Ilana Witten, who built the Cree driver rats. Kay Tai, who led the depression-related work on those rats. And then on the clarity work, Kwang-Hun Chung, who's now starting his assistant professorship at MIT, and Viviana Gradinaru, who is an assistant professor at Caltech. So just a wonderful group. We have a lot of resources online, as I mentioned, and I want to particularly thank our funding agencies, HHMI, the NIH, key support from the Gatsby Foundation, and many others. And then thank you for your time. I hope I didn't run on too long, but thanks very much.
Share the microphones, I guess. So thank you very much for that really fantastic lecture. And I think every neurophysiologist here is already generating some ideas what you can do with these really fantastic tools. And I'm very thankful that we had, as sort of the last lecture of this fantastic meeting, such a highlight in terms of techniques, how to work on the brain in the future. And with that being said, I would like to present you with a memory of this, the Wallace Fenn Lecture 2013 to Karl Deisseroth.